There's an enzyme called acetylcholine esterase that pretty much immediately breaks down the acetylcholine in the receptor site and stops the movement of those ions. And if we want to continually send a signal via this postsynaptic neuron, we have to continually stimulate that postsynaptic neuron, because the acetylcholine is going to continually be broken down. I believe there are some drugs that are given to Alzheimer's patients that render acetylcholine esterase inactive, that allows the acetylcholine to stay in the synapse longer so that those nerve signals can be generated. What? They're not showing it on here, but acetylcholine esterase would come in and break down acetylcholine. And when acetylcholine is then broken down, we're removing that ligand, and the shape of that protein then changes again, and the channel closes, and we no longer get the diffusion of those ions, and the signal stops. Or we do not generate another nerve signal. So one of the ways to stop synaptic transmission, or to stop the generation of a nerve signal in the postsynaptic cell, is to remove the neurotransmitter with an enzyme. So acetylcholine esterase is one of the ways to actually physically break down that neurotransmitter in the synaptic cleft. And your book gives a list of these methods. Degradation in synaptic cleft. That's what acetylcholine esterase does. There are some synapses that rely on reuptake. Many of the monoamines, catecholamines, undergo this reuptake process in the central nervous system. What happens is astrocytes, no, wrong one, sorry. They get brought back into the presynaptic cell, and there's an enzyme called monoamine oxidase. Yes.
What process is
This is how the synaptic transmission process stops. One of the ways is to physically break down the neurotransmitter in the synaptic cleft. That's what happens in these cholinergic synapses with acetylcholine esterase. Another way, which happens in the central nervous system, involving the monoamines, is that monoamine oxidase will break down the neurotransmitters once they're brought back into the synaptic cleft. Or sorry, back into the synaptic knob. If you ever look at the back of a bottle of over-the-counter medication or pills or something, it'll say, do not take this medication if you're taking MAOIs, monoamine oxidase inhibitors. There are drugs that inhibit monoamine oxidase, which allows those monoamine neurotransmitters to stay in the synaptic cleft longer and continue to do their job. Okay? It's a pretty powerful class of antidepressant drug. There's also things like Prozac and other selective serotonin reuptake inhibitors that prevent these neurotransmitters from getting brought back into the presynaptic cell, and therefore they can stay in the synaptic cleft longer and continue to do their job. So once they're brought in by reuptake, monoamine oxidase will break them down. And the third way that these neurotransmitters can be dealt with and synaptic transmission stop is through diffusion. And what happens there is that the neurotransmitters diffuse into the extracellular fluid. And this happens in the central nervous system. Neurotransmitters diffuse into the extracellular fluid. And it's there that astrocytes then absorb these neurotransmitters, and then they give them back to the neurons. So I mentioned the other day that one of the functions of astrocytes was to help modulate and regulate the levels of neurotransmitter in the central nervous system. This is how they do it.
What do the astrocytes do?
They're going to, after the neurotransmitters diffuse into the extracellular fluid away from the synapse, after they've done their job, they kind of diffuse out. The astrocytes will absorb these neurotransmitters and then give them back to the neurons. So we looked at a couple things. We looked at the process of synaptic transmission. What happens when that nerve signal reaches the synaptic knob, the specific movement of ions, and then we looked at how do we get that signal in the postsynaptic cell to stop being generated. And we can break down the neurotransmitter in the synaptic cleft like we do with acetylcholine and acetylcholine esterase. We can bring the neurotransmitters back into the presynaptic cell and break them down there. Or those neurotransmitters will diffuse away from the synaptic cleft into the extracellular fluid and then be picked up by other cells and then given back to those neurons. Okay? All right. I think that's everything on the learning guide for Chapter 12. There are some diseases there. E10. I think I had you add to that MS. Didn't I?
Yes.
Amber.
What did you say determines whether a synapse
The neurotransmitter and the receptors. Now what we looked at here was an excitatory cholinergic synapse. So what we just went over was an excitatory cholinergic synapse. There's inhibitory cholinergic synapses. And there's excitatory and inhibitory adrenergic synapses as well. But like I said before, we're not going to get into those specific physiological processes this semester. All right, I have to run to the restroom real quick. Coffee and water. While I'm gone, I want you to look through Chapter 12, things we talked about, and when I come back, I'll answer any questions you have about that and what we just talked about. And if you don't have any questions, start thumbing through Chapter 13, looking at the spinal cord anatomy and the spinal cord microscopic anatomy, because that's what we're going to finish up with today. All right? I know, it's almost the end of class, and I got to go now. Sorry.
Oh, really? Mind if I come down there at 1:30? Are you into some of this stuff?
How do you do that?
I'm going to probably be looking at this stuff and then we'll be asking questions on this. I might have some questions on the heart. I might have some questions also too on test for digestion.
You do. I already have plans.
Digestion. How was, do you have to test
Like,
you talk about bolus? epiglottis,
She
glottis.
gives you a paragraph, and you have to, you go along and read along with it, and it's like, food enters the bowel, the oral cavity, and it goes down to it just follows all the way to the bottom. Yeah. Bolus and time are on it, and then Rugae is on a different part.
They talk about the 3 layers of the stomach.
Yeah, it's in a slide.
Longitudinal oblique. Does she talk about the cardiac sphincter?
Yeah.
Does she talk about the paragraph?
Yep.
Does she talk about the body?
Yeah.
Does she talk about the environment?
Yeah. There's 50 questions.
Does she talk about epiglottis, degenerate?
Yep. It talks about all the.
Talk about
I still thought it
in
was.
the muscular
Yeah.
and the
Slide.
talk about the after the talk about the endocecal valve. Talk about the C-section.
Go another hour now.
No.
You have appendix, and you go to EA, something
Does this?
There used to be a bathroom right over there, and they converted it to a women's room, so I got to walk all the way over to by the elevator.
Does this sort of transmission have something directly to do with
Yes. Okay, question was asked. Does this synaptic transmission process have to do with myasthenia gravis? Yes, it does. Remember, with myasthenia gravis, it's an autoimmune condition that destroys the acetylcholine receptors. So what happens is that if there's no acetylcholine receptors, you don't get the ions moving into the cell, and the likelihood of having an action potential causing a nerve signal, well, in that case, it's muscular. And so if you don't have the neurotransmitter receptors, you don't get the movement of those ions, and you don't get an action potential in the muscle cell, which means that you don't get the release of calcium, you don't get troponin moving out of the way, and you don't get myosin binding to actin.
I mean, I remember reading that it had to do with like face or something.
It seems to manifest itself a lot in kind of the face and
But
the
this
droopy eye.
happens all over the body.
This happens all over the body. Every single skeletal muscle cell relies on an excitatory cholinergic synapse, and it also happens in the autonomic nervous system, specifically the parasympathetic. Actually, it happens in both sympathetic and parasympathetic in different places, where this process happens, where you get the movement of these ions that results in an action potential. Now, an action potential can be a nerve impulse, or it can result in skeletal muscle contraction, or cardiac muscle or smooth muscle contraction as well. So action potential just refers to a specific movement of ions in a cell. If we're talking about nerves, it results in a nerve impulse. If we're talking about muscles, it results in muscle contraction. Any other questions from Chapter 12?